DNA replication, making DNA out of nucleotides. Cells need to make copies of their DNA during S phase so that later when they divide, each of the daughter cells has a copy. In order to do that, they need to take nucleotides, which are the monomers of DNA, and string them up into a long polymer. There are several ways that scientists describe this process. One is to talk about DNA synthesis. To synthesize most nearly means to make, so during S phase, an extra copy of DNA is being synthesized. In the case of DNA, though, you're not simply making a new DNA molecule, you're copying the old DNA molecule into a new DNA molecule that has the same sequence of nucleotides inside. For that reason, another word could also be used, replicate, which most nearly means to copy. Hence the title of this talk, which is DNA replication. A final way to say this is DNA polymerization. So to polymerize means to string together or to make a polymer. So when you're making more DNA, you are polymerizing the little monomers into a polymer. Every so often you might also hear somebody say that DNA is being duplicated, though I usually save the word duplicate to talk about chromosomes when they are duplicated and they have the two sister chromatids in them. So I'll only use duplicate in that sense, and I suggest that you do the same. Better, more consistent words are either to synthesize DNA, replicate it, or polymerize it. Because DNA contains a code, when you synthesize a new copy of the code, it must be identical to the old copy. For if there were any copy errors, then the sequence over here, which actually codes for something in a cell, would no longer code for that same thing in the new DNA molecule. So how do cells do it? Well, they start first with their one parental molecule of DNA, so this is the old DNA molecule, and they split the weak hydrogen bonds that are right up the middle of the molecule. In so doing, the code which was originally closed on the inside of the helix right here becomes revealed. And now both parental strands can be used as a template or a means to copy as the new strands are being synthesized in the middle. Since thymine only hydrogen bonds with adenine, if there are a bunch of nucleotides like right in here floating around inside the cell, a free-floating thymine will line up so that it can hydrogen bond with an appropriate adenine, as an adenine would with an appropriate thymine. These base pairs right here are known as complementary base pairs, because they are always found opposite each other in the helix, and because together they make a whole. Kind of like complementary colors are found opposite each other on a color wheel. So yellow and purple are both complementary colors. Blue and orange, since they're opposites, are complementary colors. Red and green are both complementary colors because they're opposites, just like A and T are found opposite each other in the DNA helix. In math, the word complementary is also used similarly. If you have complementary angles, that means you take one angle, like this angle beta and this angle alpha, and they add together in order to make a larger full angle. In this case, complementary base pairs, A plus T, together make the two halves of the DNA helix. So these two bases, again, are called complementary. So once new complementary base pairs have been formed with both of the parent helices, you now have two new helices that have the exact same sequence on both sides of the helix as the original parent molecule. Ta-da! And as long as this happens successfully during S phase, then by the time the cell gets to prophase and gets ready to divide, and the chromatin condenses into chromosomes, we will see the two sister chromatids on each of those chromosomes, each containing their own identical sequence of DNA. Seems simple, right? Well, it's not. This gets more complicated as we start picturing things in 3 dimensions. You can see now here's the original parent helix getting pulled apart in this direction and in this direction, and then new nucleotides are added right in the middle of this structure. Like, for example, what might go right across from this T? you guessed it, an A, and then across from this A right here, a T would attach. And they don't just attach because they're in the mood to. Like with any chemical reaction, DNA replication is catalyzed or sped up by enzymes, such as this one right here. Can you guess what this enzyme does based on its name? We'll be back to that enzyme in a moment. First off, hydrogen bonds are weak, but they don't break themselves. For that we need a special enzyme. So DNA helicase, you can maybe guess from the name helicase that this is an enzyme, that acts on the DNA helix, and in this case DNA helicase breaks the hydrogen bonds between the nitrogenous bases to reveal them so that the code can be read. And if you ever meet a cute biologist that you want to flirt with, consider this pickup line. Next comes another important enzyme called DNA polymerase, which is named perfectly because it's an enzyme that makes polymers of DNA. And so after the DNA helicase comes in and breaks the hydrogen bonds and the new code is revealed, the DNA polymerase comes along, docks onto the parent strand right here, so lands on it, helps to match the free-floating nucleotides that are floating around in the nucleus with their appropriate complementary base pair on the parent DNA molecule. And then lastly, catalyzes the formation of covalent bonds between the nucleotides as they come in, so between this nucleotide and the newly forming strand. And thank God it does, because nucleotides don't just magically turn into DNA. Like in any chemical reactions, the nucleotides need to get a certain amount of activation energy before they polymerize. And that's a lot of activation energy. Luckily, if the DNA polymerase is around, the activation energy is greatly reduced just from here to here, and that makes it more likely that the DNA molecule can actually form. But while this is less energy than this, it's not no energy, so where does the energy come from? Well, a molecular diagram of DNA will help us figure that out. In this diagram of DNA synthesis, you can see the template strand, or the parent strand right here. Here's the sugar phosphate backbone in the template strand, and the bases that are coming off of each of the sugars. And then here's the newly forming strand over here, coming this way, and you have a new incoming nucleotide that's about to get added onto the end. But it's not just any old nucleotide. How many phosphate groups does it have? 1, 2, 3. 3 phosphate groups. That sounds familiar. The fancy way of saying that is triphosphate, and we know of one triphosphate, adenosine triphosphate, or ATP. We know that the cell uses ATP as its source of energy, but what we didn't really know yet is that ATP is also a component of DNA. The A is one of those A's, G's, C's, or T's. And so when a cell is synthesizing a new set of DNA and it needs a little kick of energy, all it does is take an ATP molecule which has energy stored already inside those bonds holding the phosphate groups together, and then catalyze the formation of a covalent bond between that last remaining phosphate and the previous sugar molecule, allowing a new nucleotide to be added on. In conclusion, ATP is basically the coolest molecule ever, because not only does it provide energy for the rest of the cell, but it also provides energy for its own addition to DNA molecules. So when all of this is said and done, what do we have? DNA replication produces 2 double helices. So if this was one parent double helix, then after DNA replication occurs, we've got 2 double helices, which are identical to the parent strand, and they are half old, here's the blue half, and half new, each of them. So sometimes this way of replication is referred to as semi-conservative. So to conserve means to keep the same, so semi-conservative means half of the original DNA was kept the same, but the other half became new. Half the same, half new. So DNA replication happens in a semi-conservative manner, making two identical helices from the original parent helix by splitting it apart and having it half old, half new. And this has continued since the original piece of DNA to all of the bits of DNA that exist in every single one of your cells and inside the cells of every other organism on this planet.